In this lab, we implement molecular identification technique. The barcoding marker CI gene will be used. Based on Souza et al. 2016, the mitochondrial cytochrome oxidase subunit 1 CI gene is one of the most popular markers used for molecular systematics. Fragments of these genes are often used to infer phylogenes, particularly the region near the 5' which is used by DNA barcoding consortium. We perform the DNA isolations using a PCR test. This method involves the use of enzyme, primers, buffer solutions, and thermal cycling to replicate the target DNA regions exponentially. The primer used in this lab is CI primer, and the buffer solution used is CETA. Next, we perform gel electrophoresis to separate mixtures of biomacromolecules such as DNA, RNA, and protein. The agarose gel viewed under UV transilluminator and DNA band was observed.
Pour a small amount of liquid nitrogen. Put into a mortar, grind into powder. Add 1 milliliter of CTAV into the sample and grind until they become homogeneous. Transfer 500 UL of sample into microcentrifuge tube. Incubate samples in water bath at 65 degrees Celsius for 1 hour max. Take out the sample from water bath. Add 300 UL of CIA into the sample. Mix well by slowly inverting. The tubes do not shake. Centrifuge the samples at 13.5 revolutions per minute for 15 mins. Transfer the supernatant into a new microcentrifuge tube. Add chilled isopropanol, 3/4 or 1:1 ratio into the tube. Incubate the sample overnight at 4 degrees Celsius. After incubation, centrifuge the samples at 13.5 revolutions per minute for 15 minutes. Discard the supernatant and keep the DNA pellet inside the tube. Add 1 milliliter of 70% molecular grade ethanol into the sample tubes. Centrifuge again at 13.5 revolutions per minute for 15 mins. Discard the supernatant ethanol. Make sure the DNA pellet is still inside the tube. Air dry the DNA pellets 1 hour at least under laminar. Flow at room temperature. Do not over dry the DNA pellets. Dissolve DNA pellets by adding 50 UL of sterile distilled water or sterile ultra-pure water. Perform DNA gel electrophoresis using 1% of agarose gel.
lebih atau?
Result and interpretation for DNA isolation protocol CTAP method. This is the result from our group. Sample of DNA that have been digested by enzyme are put into gel well and placed in an electrophoretic tank that have positive and negative electrode to perform electrophoresis. In DNA marker analysis, the purpose of gel electrophoresis is to separate and visualize different size DNA fragments. The initial step will be the extraction and amplification of DNA. The collected larva's DNA will be taken, and particular areas like the mitochondrial COI gene, which is frequently used to identify insect, will be amplified via PCR. After that, gel electrophoresis will be applied to the amplified DNA fragment. DNA fragments are separated by size using this procedure, with smaller fragments passing through the gel more quickly. DNA strain can be used to see the different bands formed by the separated fragments. Researchers can compare the band pattern of this sample to sample from identifiable insect species or those with non-DNA markers. It is possible to examine and contrast the past fingerprint produced by each DNA with databases of recognized insect pests. When paired with sequencing, DNA barcoding makes accurate species identification possible even in situations when morphological identification is difficult or impossible, such as in the case of damaged or larva specimen. Appropriate pest control technique can be created and put into practice as soon as the pests have been correctly recognized, possibly saving the maize crop and easing farmers' concern. From the result, there are several things that can be observed. The first one is the band present. There are visible bands in the land indicating the presence of DNA. The second one is the band intensity. The bands are unclear, suggesting that the concentration of DNA might be low. And the last one is the unclear band. The unclear of the band could be due to several factors, including low DNA concentration, incomplete staining, or insufficient exposure time under UV light. It may also suggest degradation of the DNA. So this is the result for polymerase chain reaction PCR protocol for our group. The observation from this PCR protocol is the result did not show any DNA fragment on the gel across. So, since COI primers is not being used to train the DNA, it is deposited at the end of gel as shown in the figure. So here is the comparison between our result and the expected result for PCR protocol.
There were some difficulties we faced while conducting this experiment. Firstly, pipette out the supernatant and keep the pellet inside the tube. Pellet can be hard to see, particularly if they are very small or translucent. When the pellet is small, there is a risk of losing it entirely during the process. To overcome this, carefully angle the pipette tip to the sides of the tube and gently draw the supernatant to minimize disturbance to the pellet. Next is pipetting technique. Improper pipetting technique can lead to inaccurate value measurements, cause contaminations, and affect the result. To overcome this, ensure consistent use of calibrated pipette, change tip between samples, and practice steady hand movement to maintain accuracy and prevent contaminations. Next is load and pipetting the samples to agarose gel. Loading and pipetting samples onto an agarose gel can be challenging due to the risk of spilling, air bubbles, and inconsistent sample loading, which can affect the accuracy of gel electrophoresis results. Lastly is bubble. During the extraction of the sample or protein from the tube while pipetting, the prevention of bubble is the most difficult part. The pipetting process must be done slowly and precisely to get a good result. When pipetting, avoid rapid movement and angle the pipette tip along the tube wall to reduce the introductions of air.